So, now that we've all understood that our gut is our second brain, that it's a highly exposed immune organ, that it's a nervous system of its own with 200 million neurons, let's talk about nutrition. Nutrition that's allow the enteric and central nervous system to achieve a right balance. And let's talk about algae. Algae that communicate with our bowels. So we now have a speaker who's been working with the Pierre and Marie Curie University in Paris. He is directing the biological station in Roscoff in Brittany. He specialized in the study of large marine algae. He works on the physicochemistry of polysaccharides, on marine algae, the immune system in these plants, and the knowledge of their genome. Please welcome Professor Bernard Kloareg. Professor, you have the floor. Thank you.
So I can have the first slide, please. Maybe it's for me to do it? Okay, so the title is Marine Algae for Nutraceutics from Porphyra, which is a red alga, to humans. So basically what I'm going to do is to present you what algae are, because we've been speaking of algae for quite a while this morning, but we need to explain what they are, and then basically what could be the properties of their polysaccharides in nutrition. So I'm coming from the Station Biologique de Roscoff, which is here, and as you can see, the seawater is here. Okay. But if I go to the next slide, the seawater is gone, which means that we are in a megatidal sea with an amplitude between the high tide and the low tide, which is as high as 10 meters, well, actually 9.6 meters. And what you can see here is that, of course, behind, just in front of the marine station, we have a large amount of kelps, which are laminarialis. We call them kelps. And basically, all of the ecosystems around Brittany are dominated by kelps, brown algae. Now, these resources has been used in Brittany for quite a long time already. It started by the extraction of potassium hydroxide, sorry, but also eventually about 3 centuries ago, those marine plants were used for extracting iodine. Nowadays, it's more used to extract phycocolloids, long molecules, which are gel-forming molecules. I'll detail what they are after. And we think that, so this was done by harvesting from the natural populations, including with boats. We call them guémoniers. But we believe that, in the next decade, we will have to grow to cultivate algae. This is, of course, not new. I mean, this is done largely in Asia, especially in China, in Japan, in Korea. But for Europe, it's quite new. Okay, so I've shown you brown algae, kelps, and now I am showing you red algae and green algae. So there are three main categories of multicellular algae: brown, red, and green. And you call them all algae, but in reality they are not at all the same kind of plants. So this is what we call a phylogenetic tree, which displays the relationships between the living organisms. It's based on comparisons between gene structures, phylogeny. So what you can see here, you're familiar with animals. This is a dogfish. You're familiar probably with land plants, a bit less familiar with green algae, red algae, and you may not know about brown algae. And between here, for example, this is a model brown algae we are working with, which is Ectocarpus siliculosus. Between here, two animals, you have 3 billion years of evolution. Between this alga, this brown alga, and the red alga, also 3 billion years of evolution. And between the red algae and the plants, it's about the same. Well, maybe a bit less, but not much less. So phylogenetically, green algae, brown algae, red algae, they are not at all the same. They all are plants, they all are marine plants, this is why we call them algae, but from the evolutionary point of view, they are completely different. Now, okay, red alga. So this is very important for us as we are interested in the biology of algae. This is a major asset for studying evolution of life. But what is important to know today is that they feature unique. What I mean unique is that they're completely different from the one you find on land, unique primary and secondary metabolites. Now, so I'll concentrate today my talk on the polysaccharides, not only because I'm working on them, but probably because that's the main asset for nutrition. So those polysaccharides, either from brown algae here or red algae here, they are completely different from the polysaccharides you find in land plants, such as pectins, for example. They are quite different. I mean, it's quite a high diversity. It's based on the structure of the carbon backbones, different leakages, alpha-1,3, alpha-1,4, and so on. So that already brings a layer of diversity. But then another layer of diversity is based on the substituents on that backbone. So for example, here you see a sulfate group or sulfate substituents in fucans, sulfate substituents also I come back to this, in the galactans of red algae, and here it's not sulfate, it's carboxylic substituent in alginate. So all of this, the structure of the backbone and the nature and the number and the location of the substituent make a very high diversity of polysaccharide in marine algae. For example, because I will come back to this, so this is the structure of the sulfated galactans of red algae from agarose to lambda carrageenans, and you can see that basically it's the same structure, except that in agarose you have no sulfate substituent, in kappa carrageenan you have 1, in iota carrageenan you have 2, and in lambda carrageenan you have 3 per disaccharide repeating unit. So plenty of different molecules to work with. Okay, this is more the skin, not actually the gut, but that's a structure of the extracellular matrix at the basal lamina in the skin. And sorry, what you can see, okay, this is not working, yes. Right. In the skin, the extracellular matrix of the skin and in other tissues is mainly made of proteins. In the animal, it's collagen. But you find also some polysaccharides, or actually some proteins which are substituted by polysaccharides, which we call glycosaminoglycans. This is one, perlecan. And the polysaccharides which substitute the proteins are, as you can see, also either polysaccharides with carboxylic groups or polysaccharides with sulfated groups. The one you are probably familiar with are chondroitin, dermatan, heparan, keratan. Now, this shows you that there are structural analogies between the polysaccharides of the algae, sulfated or carboxylic, and the polysaccharides of the extracellular matrix of animals. The question arises this is structural analogies, but do they have also functional analogies? Okay, I'm going too fast. In one, oh. Okay, so this is what we know about the function of the extracellular matrix in eukaryotes, especially in the multicellular eukaryotes. Okay, of course, the first function of the extracellular matrix is tissue integrity. If you remove the extracellular matrix, the cells, this is plant cells, they are becoming isolated round protoplasts, as we say here. But they are also, the extracellular matrix is also involved with cell differentiation and development, with cell expansion, with cell-to-cell adhesion, with signalization, and of course, with interfacing with the environment. So the extracellular matrix, hence the component of the extracellular matrix, including the polysaccharides, are involved with this function. So we've been studying this in algae too, and I'll give you only one example but there are many examples, and it's the same in algae too. So basically what we show here is, okay, this is ultraviolet picture, and this is Ectocarpus siliculosus, the brown alga. No, it's an Ectocarpus. So it's not Ectocarpus siliculosus, it's Ectocarpus subulatus, which is an Ectocarpus strain which is able to grow, actually which colonizes fresh waters, but which is also able to grow, or at least to survive, in seawater. So what we've been doing is we've taken this species, leave it in seawater, no, leave it in fresh water, put it in seawater, and then stained the alga by a monoclonal antibody, which is specific for sulfated fucans. And what you can see is that when this alga is maintained in fresh water, it doesn't make sulfated fucans. When it is put or cultured in the presence of seawater, it does make a lot of sulfated fucans. And we know that sulfated fucans are highly hygroscopic polysaccharides. They also have 3 sulfate group per disaccharide. So they are probably involved with ionic and hydric regulation in this algae. And as you can see, this is only expressed when the alga is cultured in seawater. Okay, so this is first slide summarizing what I've been saying. You have a high variety of polysaccharides in algae, with the exception of cellulose. And the skeletal polysaccharide of the matrix is always, or most of the time, cellulose. That's always everywhere, including in land plants. But when you come to matrix polysaccharides, you find either, okay, in the brown you find sulfated, well, a lot of sulfated polysaccharide, alginates. In the reds you find carrageenans, agar, porphyrin. In the green, something which is quite important for Olmix because it's using this polysaccharides a lot, pulvans. And as I said, so they have evolved a high diversity of matrix extracellular polysaccharides. They are, okay, this polysaccharide you have to remember, well, you have to understand that they are ancestral components of the extracellular matrix. They are present in animals and in algae, which means that they derive from very ancestral molecules. But they are not present in land plants. And they're not present in land plants because land plants do not have much sulfate in their environment. It's only several micromolar in the interstitial seawater, whereas in seawater it's 20 millimolar. So they're not in land plants. So, and I showed you that, so they are homologues. They derive from the same molecules, and they are functional analogues of the matrix of the glycosaminoglycans in animals. Functional analogues, that means that in algae, they share the same function as the glycosaminoglycan do in animals. The question then is, would the sulfated polysaccharides of algae be recognized one way or the other by animals? Even though animals and algae have, 3 billion years of evolution, basically, between themselves. Okay, so when you look at a review, and this is a quite recent one, of the bioactive compounds you find in seaweeds, you of course always find polysaccharides coming from either brown algae, red algae, or even green algae. There are other active compounds which are secondary metabolites, for example, but polysaccharides always come in a good place. And actually, when you read the literature also, those polysaccharides are quite active in various domains, antiviral, anti-inflammatory. I mean, you can read it as much as I do. A lot of activities have been described in vitro. Some in vivo, but not that many. So what I think, it's an hypothesis, of course, but I think that these properties of the algal polysaccharides are probably related to the structural and functional relatedness of polysaccharide, well, extracellular matrix polysaccharides in animals and marine macroalgae. we see whether it's true or not. Now I'm switching to the second part of my talk, which is how can you get those polysaccharides out of the algae? So you can, of course, extract them by chemical methods, but it's probably wiser to try to extract them and process them by using enzymes that are specific for the degradation of those polysaccharides. Now, why is it better to do so? Because, well, enzymatic methods are usually more specific and more reproductive than chemical methods such as acid hydrolysis or even other kind of hydrolysis. Now, by using enzymes which degrade the polysaccharide of algae, you have a lot of other things you can do. I mean, you can use them as enzymatic tools to crack the algal biomass, for example, and also just to process them. Now, so something you need to remember is the polysaccharides from seaweeds are very different from the land plant polysaccharides. So it's clearly useless to try to use terrestrial enzyme to work with algal polysaccharides because they don't recognize them. So the solution actually is to find specific enzymes from marine organisms that would degrade the algal biomass. So we have been doing this for quite a number of years already. And actually, I'm showing you an example which was published by P, who some of you know, a few years ago. And this is what we call the classical traditional methods. You try to look for a bacterium that degrade ulva. In that case, it was ulva lactica. And this is, by the way, the primary structure. Okay, I'm blocked. Good. So this is, the bacteria isolated and shown on agar. This is to show that this bacterium, Persicivirga ulvanivorans, was actually capable of degrading ulvan. And then what she did is she purified the protein, cloned the gene, put that into E. coli, as I said, and demonstrated that it was active on ulvan. I mean, most of the time, that's the only method you can use because there is no reference on the protein structure available in any genome. But today, once you have that capacity, that category of reference, you can go, we have been doing using these traditional methods, we've been isolating a number of enzymes which degrade the polysaccharide of algae, agarases, carraginases, fucanases, alginate-lyases. We've been looking at the structure of these genes. We actually went all the way to study the three-dimensional structure. And so those data can be used as a reference to find other enzymes, other related enzymes in the genomes. Okay, so we know the genomes of algae. Actually, I'm not going to detail that. But now what we can do is to look at the genomes of the bacteria that degrade the algal biomass. And we've been working ourselves in basically with this family, the Bacteroidetes. So that's a name which is reminiscent to something to you, probably, Zobelia galactivorans, but also other categories of phyla, planktomycetes, for example, which are bacteria which are involved in the biodegradation of the algal biomass. So what we do is, since we have those references, we look into the genomes of marine bacteria. We find sequences which are probably involved with the degradation of algal polysaccharides. We study we clone them into E. coli, we express them. We look at the biochemical function of these enzymes. We can actually crystallize them, make the structure. We could stop here as far as oligomix is concerned, but we are more interested in doing other things, so we go also all the way to basically investigate the way those bacteria function to degrade the algal biomass. So we've been doing, so now there are more and more data, thanks basically also to the gut metagenomes. There are more and more data about bacteria in the literature, about bacterial genome in the literature, so you can do more and more things. And so what we've been doing, for example, is to, okay, this is the family 16 of glycosyl hydrolysis, so enzymes that degrade carbohydrate. And so we have been studying all of this. And in the genome of Zobelia galactanivorans, we had all of these sequences, which obviously were, bioinformatics still uses, are glycohydrolase from the family 16, but we didn't have any clue on which structure it was. Well, they likely were linked to agarases. So we've been studying those, actually 2 of them, to try to characterize their structure. Their structure and their function. So what we've been doing is we've been taking those 2 proteins, express them in E. coli, take the protein and try to to determine its enzyme activity. First thing we did was to try on algae. So we tried on Porphyra, Gracilaria, and Gelidium. Those 3 red algae, they do produce more and more agarose, when you go this way, and more porphyrin when you go this way. More porphyrin here, more agarose here. But this is basically, porphyrin is the same structure as agarose, except that it is sulfated here. And what you show doing this way is that, something is missing, maybe. Yeah, anyway, what we did show is that the enzyme which we have been trying to look for the sequence were not agarases, but porphyrinases, which means enzymes that degrade sulfated agar. And then we look into the literature whether these porphyrinases, enzymes that degrade porphyrin, were to be found somewhere else. And what we found is that, of course, they were found mainly in marine bacteria. But they also were found in the metagenomes of Japanese people, but not of Europeans or Americans. And so, of course, what we deduced is that probably they are found because of the dietary regime of Japanese in the Japanese people, because they eat Porphyra. Porphyra, which is named nori, and which is used to make, I said sushi here, but I went to a Japanese restaurant a few days ago, and it's not sushi, it's maki, sorry. That is a clear illustration of what Hervé said, is that actually there is your microbiota depends on your dietary regime. But for us, it's something more, because this shows a clear link between marine bacteria, let's say marine red algae, Porphyra, with marine bacteria that degrade those marine algae, with man. So it's a clear, it's starting to bridge a link between marine algae, you and I. Well, actually, especially Japanese, so if there are Japanese in the room, it's much better, just raise your hand. With Europeans, don't do that, but you do it. Now, actually, we look in more detail in the genome of Bacteroidetes plebeius, and we actually found 6 other genes which are conserved in the genome of this marine bacterium, of this gut bacterium, which are probably what we call a horizontal gene transfer between the marine bacterium and the gut bacterium. The question now then arises is, do the product of this gut bacterium, are they actives on human? So we are not as good as they are. So we actually, in terms of humans, algae, I wouldn't say. But in terms of humans, we only have been using a very simple test, which is, because that can be done in any lab, the tissue repair bioassay, and what what you do is you take skin keratinocytes, incubate them in the presence of the products that you want to test, and then ask, okay, is this doing anything? Okay, and what we have found is that the degradation products of porphyrin, the porphyrin oligosaccharides, actually are active on humans. So listening to what I've listened this morning, it is clear that you can now imagine two kinds of activities between the algal products or the algal natural compounds and humans. First, you could imagine that there is a direct activity between the product, well, let's say the polysaccharide product, and humans. But it is also very likely that there is a probiotic activity between those polysaccharides and the human microbiome. So there are two ways of acting in nutrition here, either directly or indirectly. It doesn't matter really, because what counts is the result. To finish on this, to conclude, I just wanted to tell you that this, of course, brings us a lot of optimism to find other properties in nutrition on the algal polysaccharides or algal products in general, either polysaccharides or proteins. So we are engaging into a large-scale research program thanks to the Banque Publique d'Investissement. So I'm happy that Christian Briand is here. Okay. So basically what we are doing, what we have in mind is to go from the cultivation of microalgae. This is not actually in this program. It has to be done somewhere else. But then we go all the way from bioprocessing, including enzymes, with bioassays on animal models in farms. And, well, no, not in farms, in vitro, but then going to the real condition. So I don't know whether 1 year will be enough to give you many more results, so whether I can report on this next year, but probably 2 years from now we can give you more results. So for us it's a kind of a new paradigm which really brings a lot of interesting things to do on algal products and nutrition. So I hope that we will fulfill the recommendation of the former speakers and try to go further in this direction. Thank you for your attention.
Merci, professeur. Quelques images. Même punition que les autres.
Je suis
Voilà.
While today priority is still given to chemistry, to antibiotics in particular, from now on we know that algae extracts can have a favourable influence on our digestive ecosystem. Algae has a probiotic effect, encouraging the development of such benign microorganisms. It's been proven that algae extracts can stimulate certain receptors in the enteric central nervous system. The stimuli are transmitted to the central nervous system and can influence a number of neuronal activities: our emotions, our desires. The enteric immune system's receptors, dendritic cells, can be activated by algae extracts, which they fix, then transmit to lymphatic ganglions. This will help to generate defensive immune cells, lymphocytes. These cells will then be diffused throughout the organism to defend against potential invaders, like harmful bacteria, by phagocytosis.
Voilà comment les algues influencent notre ventre. Merci, professeur.
So the algae has an influence on our gut. There we go. Thank you very much, Professor. Maybe any questions for Professor Kloareg? Yep. The question down there. We'll try and give you a mic. I won't throw it. how much I'd love to. So we saw there were enzymes used with certain bacteria. Have you studied at any point the enzymes that come from the microflora of some plant-eating fish or things such as manatees or other plant-eating aquatic mammals?
We have not been studying those enzymes ourselves. I mean, there are publications, of course, showing that. And actually in fish, I mean, some fish, I mean, we know that they degrade alginate. We actually are not working with fish, but we are working with haliotis, which are gastropods, okay? Well, I mean, mollusks at least. So we are not working on fish, but you have plenty of enzymes in the bacteria. I mean, in the free bacteria anyway, so you don't, I mean, Hervé was saying me that we could actually go and look into the human microflora to look for them, but they are everywhere, so we need to concentrate. Now, I just want to add something. I mean, before we demonstrate that algae works on neuron, I mean, we still have some work to do, right? I mean, the last explanation was going a bit fast. We show they are recognized. The delineation of the mechanisms still remains to be done.
Ok, merci professeur. Vous pouvez regagner votre place, on peut l'applaudir.
Thank you very much. You can go and sit down.